Hi everyone, here we are with another video in the Learn With Me series of the lesson Molecular Basis of Inheritance. So we have already discussed about the introduction, what is DNA, what it is made up of, what is its length, and what is a monomer nucleotide consist of, what are the various bonds present in them, and what this structure implies. All these we have discussed already in detail. So let us move on to the next topic. What is the difference between DNA and RNA? So here it is clearly explained what makes RNA. RNA has ribose sugar which has OH group at 2' position. DNA has deoxyribose sugar which has H in the 2' position. And RNA has uracil instead of thymine, and DNA has thymine, it doesn't have uracil. So these are the two major differences, structural differences. Remember, these two are the structural differences. So what are the structural differences between DNA and RNA? RNA has a ribose sugar which has hydroxyl group at 2' position. DNA has deoxyribose sugar. The sugar molecule has H at the 2' position. And RNA has uracil instead of thymine. DNA has thymine. These are the two structural differences. So what is the chemical name of thymine? 5-methyl uracil is the chemical name of thymine. Okay, so DNA basically is an acidic substance. So from the name itself we can understand DNA is deoxyribonucleic acid. So it is acidic in nature. Is that clear? Okay, who identified that DNA first? So initially this was identified by Frederick Miescher in the year 1869. Who identified it? Frederick Miescher. Which year? 1869. What is the name given by him? Nuclein. So, is it clear? So, due to technical limitation, he was not able to isolate and elucidate the structure of DNA. So, in the year 1953, James Watson and Francis Crick proposed the famous and simple double helical structure of DNA using the data obtained by X-ray diffraction technique performed by Maurice Wilkins and Rosalind Franklin. So how many scientists' name we have come across in this paragraph? 5 scientists' name we have come across. The first one, the person who identified nuclein. Okay, he is Frederick Miescher. In the year 1869, he has identified nuclein, that is the nucleic acid. Okay, what is the name given by him? Nuclein. And later in the year 1953, the structure, double helical structure of DNA was proposed by James Watson and Francis Crick. They used the data of X-ray diffraction technique performed by Maurice Wilkins and Rosalind Franklin. So the structure of the DNA is actually given by James Watson and Francis Crick, but they have taken the prime support of Maurice Wilkins and Rosalind Franklin from their X-ray diffraction data. Okay, so apart from this, one more person has also contributed in elucidating the structure of DNA, that is Erwin Chargaff. He proposed in a double-stranded DNA, the ratio between adenine and thymine and guanine and cytosine are constant and equals 1. Which also means the number of adenine or the molar concentration of adenine is always equals to the molar concentration of thymine. So in very simple words, we say that number of adenine equals to number of thymine in a DNA molecule. DNA is always double stranded. Okay, if in one strand adenine is there, another strand thymine is going to be present. They both are going to be connected by two hydrogen bonds. Adenine always base pairs with thymine, and vice versa is also true. What is meant by vice versa? The opposite of that. What is the opposite of that? Thymine also always base pairs with adenine. Is that clear? The same way, guanine and cytosine, they are going to pair up with each other by 3 hydrogen bond. What exists between adenine and thymine is a double bond. What exists between guanine and cytosine is a triple bond. Okay, 3 hydrogen bonds here and 2 hydrogen bonds in this case. So their number or concentration in case of adenine and thymine is going to be equal, and guanine and cytosine is going to be equal. When they are equal, if you are taking the ratio between guanine and cytosine, since the number or concentration of them are equal, their ratio is going to be always 1. The same applies here. Is it clear? So in other words, let me give an example how to understand this statement. Say in a DNA molecule, there are 1500 adenine bases. Okay, so that means every adenine is going to base pair with a thymine, which means 1500 thymine are there. So the concentration of adenine and thymine are equal. Okay, so when you take a ratio of concentration of adenine with thymine, 1500 divided by 1500, which is going to be 1. So the ratio is always going to be 1. The same applies between guanine and cytosine. Okay, coming to the next one. This base pair confers a unique property of the polynucleotide chain. They are said to be complementary to each other. So this is very important. The base pairing rule, what is given by Erwin Chargaff, confers a unique character. What is that unique character conferred by Erwin Chargaff? Complementary of the strand. Okay, if you know the nucleotide sequence in one DNA strand, based on the complementarity rule, you will be able to write the sequence of the other strand. So this is the basic of replication. Okay, replication happens in this manner. Even transcription happens based on the complementarity rules only. Is that clear? Okay, so that is what has been explained here. So let us move on to the next one. So what are the salient features of the double helical structure of the DNA? Okay, so this is very important. Even though it is very basic, some students tend to make mistakes in this area. What they actually do is, every point has a complete thing, but sometimes students, what they tend to do is they break the point so that the concept becomes incomplete. They break the point, write that as a 2 or 3 points. It's okay, but altogether that is going to make 1 point only, however you break it. Okay, so remember that, learn the concept completely. It's very important. Okay, so the first point here, it says that there are 2 polynucleotide chains where the backbone is constituted by sugar phosphate and the bases project inside. So the first point is very simple. It talks about the double helical structure. It says there are 2 polynucleotide chains. Each of them have sugar phosphate as the backbone and the bases are projecting inside. Okay, so now the next one, the 2 chains have anti-parallel polarity. One chain is going to be in 5' to 3' position, other chain is going to be 3' to 5'. Already we have discussed about polarity in the diagram already. So here we have discussed, this is the 5' end, this is the 3' end. 5' is phosphate end, 3' OH end. So in this is the backbone, the bases are projecting here. So if the bases have to be projected inside like this, other strand must be in the opposite anti-parallel direction. Hence it is going to be 3' here and 5' this side for the other strand. That is what has been explained by this point. Okay, the bases in the two strands are paired through hydrogen bond forming base pairs. This we have already discussed with the Arabin-Chargaff rule. Adenine forms two hydrogen bonds with thymine from opposite strand and vice versa. Similarly, guanine is bonded with cytosine with 3 hydrogen bond. And very important to note this one, purine always base pairs with the pyrimidine. So, in one strand there is going to be purine, in another strand it is going to be pyrimidine, and vice versa. So, always purine base pairs with pyrimidine. That you never forget. Okay, this generate approximately uniform distance between the two strands of the helix. So, how come this generates uniform distance between two strands of this helix? Okay, if you remember the purine structure, it is a single heterocyclic molecule. Okay, it is like a similar, so we can say that it is like a benzene ring, similar to that. Okay, a ring structure, just one ring structure is present, but in case of pyrimidine there are going to be two ring structures. So just having one ring, let me draw it here, just having one ring like this, and another one is going to be having two ring like this. Okay, so sorry for the very bad diagram, but it is for your understanding. So this is the distance between them, this is the distance between them. So if it is going to be just one ring like this in both the cases, the distance is going to be smaller because it has to reach it, so it is going to be smaller. So to maintain the distance between the two strands, the gap between the two strands is maintained by this base pairing property, that is purine always base pairs with pyrimidine and pyrimidine always base pairs with the purine. So what are the purine bases? We use the term PAG. Purine bases are adenine and guanine, and pyrimidine bases are cytosine, uracil and thymine. PCUT, that is a term we have used to learn. So now coming to the next point, the two chains are coiled in a right-handed fashion. So what is meant by right-handed fashion? So they are going around in a right-handed manner. So they are moving towards right hand. So the pitch of the helix is 3.4 nanometer and there are roughly 10 base pairs in each. So let us understand this by looking at the diagram. Okay, so this is the double helical structure of the DNA and this is going to be the pitch. Okay, so this is going to be one pitch, that is another, this gap here. So this gap here is referred as minor groove, this is referred as major groove. So there are going to be 10 bases in each helix, each turns. Okay, so this makes the distance between a single base pair is going to be 0.34 nanometer. Okay, so the pitch is made up of 3.4 nanometer and roughly there are 10 base pairs. So 10 base pairs covers a distance of 3.4 nanometer. Hence one base pair, the distance between one to the next base is 0.34 nanometer. Okay, this is a simple logical math. Let's move on to the next one. So here it is explained. The plane of one base pair stacks over the other in the double helix. This, in addition to hydrogen bonds, confers stability of the double helical structure. So this is an important 2-mark question, this fifth point. So in addition to the hydrogen bond between the base pairs, the stacking of one base pair over the other confers stability. So this is how the stacking is done. To understand this better, let us look at this diagram. So one base pair, the next one, the next one. So the way they are stacked one above the other confers stability. So two things are conferring stability. What are they? The hydrogen bonds between the bases of the complementary strands and the stacking of one base pair over the other. So these two are conferring the stability. So how many salient features of the DNA are there? There are 5 features. In the first feature, we discussed about the backbone, number of polynucleotide chains. The backbone is sugar phosphate and the bases are projecting inside. In the second one, we discussed the 2 polynucleotide chains are anti-parallel. One is in the direction of 3' to 5', other one in the direction of 5' to 3'. The third one, we discussed about the number of hydrogen bonds. Adenine and thymine has 2 hydrogen bonds between them, and cytosine and guanine has 3. That is what we have discussed. And here they are coiled around in the right handed fashion. A pitch has a length of 3.4 nanometer, and each pitch has 10 base pairs, hence the distance between each base pair is 0.34 nanometer. The last point is about stability. In addition to hydrogen bonds, the stacking of one base pair over the other confers stability. So to support the polarity, let us look at this diagram. So this is a diagram of a DNA. You might be asked to draw this diagram in the exam. Okay, so if you compare this diagram with the previous one with a single strand, the same thing you have drawn as a mirror image. You can see that. Okay, you can see what you have drawn here, the same thing you have drawn here, but like a mirror image, kind of, not exactly. Okay, similar to that. Okay, here you can see that here is the 5' N, 5' phosphate group, here is the 3' OH group, and this is one strand, this is another strand. The second property we have learnt there is anti-parallel nature. What is the anti-parallel nature? One strand is in the 5' to 3' position, other strand is 3' to 5' position. That is what is anti-parallel. Okay, so just practice the diagram. Well, this is very important. Don't forget to practice many times. Okay, so if you practice this diagram, you can draw both DNA and RNA in the same way. So only thing you have to notice is what is being here in the 2' position. If you are writing it over there, make sure if it is a RNA, you write OH in this position. So in this position, otherwise you have to write H. Okay, in case of DNA. That's very simple. This is quite easy. If you want to score well, no, just follow me whatever I am saying. You will definitely score very well. Okay. So this is a diagram which will make us to understand the double helical structure of the DNA. It is a schematic diagram. Schematic diagram means a diagram which resembles the real thing in a very simplified format. Okay, so now the next one. This is the central dogma of molecular biology. So this is extremely important. This marks the basic of everything you are learning. So whatever it is given here, that is what you are going to learn in detail in this lesson. The entire field of molecular biology and genetics is actually based on this only. Okay, so we know that the genetic material is DNA in most of the organism. DNA has the capability to make its own copy. That process is referred as replication. Replication is a process in which DNA can make a copy of itself using one of the strand as a template. Okay, the next one, DNA in the process of making protein, it involves in the first step is it involves in the making of RNA. So DNA is used as a template to make RNA. So whatever the information is here, it will be transformed in the form of RNA. So the information is carried by a messenger RNA. So in addition to that, tRNA and rRNA are also formed. So every RNA is formed by the process transcription. So whatever information is in DNA is transcribed into RNA, and whatever information is present in the language of nucleic acid, these two are nucleic acid, is translated into the language of protein, which are made up of amino acid. Hence, this process is referred as translation. So, this is entirely a different component. DNA and RNA are entirely different from protein. Okay, that is why this process is named as translation. So, there are three major processes involved. DNA can copy its own self, that is replication. The information in the DNA is transcribed into RNA. The information in the RNA is translated into protein. So, three processes are replication, transcription, and translation. So, one more process is usually mentioned along with this in the later stages after the discovery of the enzyme reverse transcriptase in viruses such as HIV. Okay, in that only RNA is present. RNA will be converted into DNA with the help of reverse transcriptase enzyme, and the process is referred as reverse transcription. This is the basis of infection of retroviruses. So with this we covered up the first introductory part of the molecular basis of inheritance lesson. Even we covered the basic features, the salient features of DNA. Okay, hope this is going to be helpful. Please take a moment to subscribe the channel and like the video. If you like it, obviously, this will be helping me and encouraging me to make more such videos. Thank you.